Thank you, Charlie. The great honor of introducing speakers for today's postdoctoral spotlight. First to speak will be Dr. Kyle Card, who earned his PhD with my longtime colleague and friend Richard Lenski at Michigan State, where he studied the effect of genetic background on antibiotic resistance evolution. He is now an HHMI Hannah Gray postdoctoral fellow in Jacob Scott's lab at the Cleveland Clinic. Here, he is expanding on his previous work to better understand how demographic and ecological factors interact with background to influence this medically relevant phenomenon. He has the goal of leveraging this knowledge to control pathogen populations through rational drug therapy. So Kyle, floor is yours.
Thanks, Joe, for the introduction. Also, thank you for the nomination, and thank you to the organizers of this Club EvMed for selecting me to give a talk. So today I'm going to be discussing my postdoc work that's centered around this notion that the evolution of antibiotic resistance is a multifaceted phenomenon. So as we all know, antibiotic resistance is a serious and growing public health threat. In the United States, about 3 million individuals will contract a resistant infection every year. And of those 3 million individuals, about 36,000 will die from their infections. And the problem is getting worse over time. So it's projected that by the year 2050, about 10 million individuals globally will die on an annual basis from drug-resistant infections. And these deaths will outpace those of cancer. So given this severe and growing public health threat, I aim to understand the evolutionary processes and factors that drive this phenomenon. So evolutionary potential for resistance may depend upon several factors. As a graduate student in Rich Lenski's lab, I found that genetic background can affect both the phenotypic and genotypic evolution of drug resistance. But we too know that population size and mutation rates can influence this public health phenomenon. So as a postdoc, I want to address this motivating question, and that is, how do these factors interact to influence evolutionary paths to resistance? So population size may influence resistance evolution. So let's imagine that we have two populations, one that's of large size and the other that's small size. And let's imagine they also have the same point mutation rate. So given that the mutation supply rate is proportional to the product of the population size and mutation rate, we would expect that on balance, large populations should be more evolvable because they are more likely to sample rare mutations that provide high levels of resistance. But there are several confounding factors that might impinge upon this expectation. So these populations might have potential differences in mutation rates, and they also might have potential differences in genetic background, just to name a few. So I'm aiming to disentangle the effects of these factors on resistance evolvability from both a theoretical approach and also an experimental approach using populations of bacteria. During this talk, I will be discussing this theory approach that me and a collaborator in our lab, Dr. Jeff Altus, is working on. So Jeff and I, we designed these simulations in silico where we evolve populations from different initial genotypes on an adaptive landscape. And in the initial phase of this experiment, we evolve these populations under no selection. So in effect, these populations are exploring their landscape due to genetic drift alone. And I'm showing a representative example of two populations, A and B. And we evolved genotypes for 500 generations. We then generated a drug landscape where we can tune the topography of this landscape by changing a certain parameter that influences the degree of epistasis of the landscape. So thereby we have regions of the landscape of lower fitness and regions with higher fitness. We then transfer offspring from this initial evolution, this initial neutral evolution phase into a drug landscape, and then we measure evolvability. So given this, we know that we can see that altering population size and mutation rate parameters can lead to different outcomes. So if we start with, for example, with a large population with a high mutation rate, then the populations can explore more of the mutational space. So in this schematic illustration, population A starts close to the global optimum. The initial genotype is shown as an open circle, and the offspring of this population found the global optimum after evolving, and that offspring is shown with an asterisk. However, decreased mutation rate and/or population size can lead to less exploration of the sequence space. In this example, population A did not find the optimum. On the other hand, offspring from population B now reach a higher resistance level compared to offspring from population A. So we can see that where the population starts off on a landscape, and also its mutation rate and population size can affect evolutionary outcomes. So I also want to sort of state what I mean by evolvability in this case. So we operationally define it as the fitness difference between an individual's final genotype and the population's initial genotype. So we first looked at a fitness landscape with 32 initial possible genotypes. And we looked at evolving populations on this landscape under 5 different mutation rates and 5 different population sizes. And I'm showing here average evolvability of these 32 initial genotypes by the color shade in this heat map. So darker colors, squares equal higher average evolvability. So we see that in this 32 initial genotype landscape, the average evolvability increases with population size and mutation rate as expected, just given what I described previously with the equation for the mutation supply rate. We also see this trend across different landscapes with increasing initial genotypes from 32 to 1,024. In a separate experiment, we then evolved populations from these different initial genotypes as before. And then we found the most evolvable offspring from each population. So in this population A, in the schematic illustration, it started at initial genotype of 4 and it ended at 7. So the evolvability in this case is 3. In population B, likewise, the evolvability is 4. So what we did was we subtracted these values, 3 from 4, to determine the effect of studying genotype on evolvability. So this gives us some understanding of how genetic background or how studying genotype affects evolutionary outcomes. So we found that the effect of the studying genotype becomes more pronounced when population sizes are small and genotypic space is large. So again, at the smallest landscape of 32 initial genotypes, the maximum evolvability was highest at the highest mutation rate and population size, but this shifts to in the larger landscape to populations of small size and high mutation rate. So to sort of the interpretation of this, I'm going to show here. So I'm just showing a representative schematic illustration of the landscape with 1,024 possible initial genotypes. I'm also showing two populations on this landscape. The initial genotype of the populations are shown in these gray circles, and 10 offspring after some evolution. They've explored the mutational neighborhoods around this initial genotype and showing these offspring as teal circles. So we can see, based upon this illustration, that a population with 10 individuals, again, where we saw the maximum evolvability in this case, they will explore about 1% of all possible genotypes on average. Also remember that each genotype is associated with a fitness value. Therefore, differences in evolvability should be more pronounced in small populations because individuals may end up at more extreme fitness values due to chance. So this work and future work with experimental populations of bacteria, which again, I won't be able to talk to you all about in this presentation due to time, but I'm happy to talk about in the extended discussion section. This work gets at this question of how predictable is resistance evolution, or if we could better understand the factors that underlie this phenomenon, we therefore might be better equipped to treat resistant infections through the use of rational drug therapies. So with that, I want to make a couple of acknowledgements. Firstly, I want to acknowledge my postdoc advisor, Dr. Jacob Scott. I want to again acknowledge my collaborator on this project, the postdoc in our lab, Dr. Jeff Maltus. I also want to thank Theory Division, our lab in general, and I also want to thank HHMI through a Hannah Grace Fellowship. And with that, I'm pleased to take any questions.
Yeah, we should have time for 1 or 2 questions for Dr. Card. All right, so I'm looking in the chat and I see one question from Richard Katz. And he asked the question, Kyle, does this model take into account horizontal gene transfer, which I imagine would increase with population size?
Oh, yeah. No, it doesn't take into account HTT yet. It's something I want to look at in the future. And I also agree with that expectation that with increase in population size, one would expect an increased rate of HTT. That's really a good insight and something I want to look at in the future.
All right. Are there any more questions for Dr. Card?
Kyle, I'd be curious just to hear a little bit more about the empirical work, if you could say a few words about the systems you used or anything like that.
Yeah, so I want to look at these questions using clones that have been isolated from Richard Lenski's long-term evolution experiment, particularly in a population that evolved increased population size after the evolution of a novel metabolic trait, and then it subsequently evolved increased mutation rate. And of course, across this evolutionary history, it has differences in genetic background. So through careful manipulation of the experiment, we can isolate the effects of background, population size, and mutation rate on resistance evolvability, and then look at their combined effects. So that's, I have a couple of slides if anyone wants to ask me any more in-depth questions in the discussion section afterwards. I'd be more than happy to to discuss my plans.
Yeah, that's great. And that's just a reminder that we will have breakout sessions with each of the speakers at the end. And so we could continue that conversation there.
All right. Thank you, Kyle. So our next speaker in this session is Dr. Liz Lang, who is a postdoctoral associate in the Susan Alpert's lab at Duke University. Her research focuses on how socio-environmental factors during early and adult life affect phenotypes and fitness. Liz received her BS in biology and psychology with a minor in animal behavior at Canisius College before completing an MS at Clemson University and a PhD in biological science at Florida State. Liz, take it away.
Thanks, Joe, and thanks all for the invitation and for coming to the talk. Today I'm going to tell you about some of my postdoc research linking the effects of early life experiences, adult social bonds, and lifespan in wild baboons. So we know that early life experiences are important. For example, from many studies in humans, we know that adverse childhood experiences like abuse, neglect, and household dysfunction can negatively affect developmental processes. And ultimately, this has been shown to lead to negative behaviors in health in adulthood. And of course, these processes don't just occur in humans. We see that early life experiences are important and have lasting effects on fitness and other phenotypes in many different animal systems. However, it's unclear how experiences during early life and adulthood translate into differences in lifespan. So in the baboon system that I work on, we have evidence for parts of the links between early life adversities, adult traits, and fitness. For example, we know that individuals who experience more negative conditions during early life have decreased lifespan. And we also know that individuals who experience more early life adversities have decreased bonds with other females, as well as decreased social bonds with males. And we know that adult traits are important in determining fitness. For example, individuals who have weaker social bonds with males and females have tend to live less long. And so the obvious hypothesis here is that social bonds mediate this relationship between early life adversity and fitness, where individuals who experience more adversities have reduced social bonds and therefore decreased survival. However, we have to actually test that hypothesis empirically. So for example, we don't know if adversities early in life affect fitness directly, as shown by this green arrow, or if there's this mediation effect where early life adversity affects adult social bonds, and then adult social bonds affect fitness. So today I'm going to tell you about some of my work where I ask if social bonds mediate this relationship of early life adversity on survival. And in addition, I'm going to tell you about some work where we ask if they moderate this relationship. And so in other words, I'm asking if individuals have early life adversities, can they overcome this by strengthening their bonds with other individuals in the population? And to do this, we took advantage of a data set from the Amboseli Baboon Research Project. And baboons in this system are located in southern Kenya and northern Tanzania. And this project is led by Susan Alperts, Jenny Tang, Beth Archie, and Jean Altman. And data have been collected on over 2,000 individual wild baboons living in free-ranging social groups continuously since 1971. And these data are collected by experienced observers and with help from support staff pictured here. And so I wanted to take a moment to acknowledge Kinua, Raphael, Langita, and Sarah, who have collected all of the data that I'm going to be talking about today. And they recognize the individual baboons by sight and are currently collecting data on about 300 living individuals across 5 social groups. Now, Chelsea Weibel and Beth Archie provided a great introduction to the system last week, but if you weren't here, the baboons live in mixed sex social groups of 20 to 150 individuals, and males disperse at maturation, while females remain in their maternal social group. They're a good model for human life's effects on human health because development is proportional to humans, but accelerated. So that allows us to measure full life courses on these individuals. And from this research project, we've compiled a data set of 199 females, and from them we have over 1,800 years of life represented that we use to measure social bond strength, and 74 deaths that we use to measure survival outcomes. And we're only modeling female survival because males disperse from their natal groups, as I said, and so that makes estimating survival a bit more complicated. So I'm just going to tell you about females today. Our measurement of early life adversity is this cumulative adversity index, which is the sum of 6 sources of early adversity. 2 have to do with resources, so whether individuals experience drought in their first year or were born to a large group. 2 have to do with their mother's social environment, so if their mother is of low social status or is lowly socially connected to other individuals. And 2 have to do with family structure, so if they lose their mother before they reach sexual maturity, or if they have a close in age younger sibling. And our measure of adult phenotype that I'm going to talk about today is social bond strength, which we calculate using the dyadic sociality index. And this measures social bond strength of a female's top social partners based on grooming relationships. And grooming is a really important affiliative behavior in primates, including baboons. And we calculate a dyadic index for both females' relationships with males and females' relationships with other females separately, because relationships with males and females tend to have different relationships with fitness and effects of early adversity. And here, lower values mean that an individual has weaker social bonds than the population average, whereas higher values mean she has stronger social bonds with her top partners than the population average. So in order to test our hypotheses, we're using mediation analysis with joint modeling. And this approach is being developed by 2 Duke statisticians, Dr. Fan Li and Dr. Shuxi Zhang. And allows us to take into account the longitudinal nature of our social bonds and examine if those are the black box mechanism that mediate the relationship between early life adversity and survival. And so when I say mediation effect, I mean that we're testing that if part of the reason that early life adversity affects survival is because early life adversity affects social bonds, which in turn affect survival. And this analysis also allows us to estimate parts of this framework. So we determine the effects of cumulative adversity on social bonds, shown in orange, and also determine the effects of social bond strength on survival, independent of changing levels of cumulative adversity. We then, of course, estimate the total effect of adversity on lifespan. And this total effect can be broken down into 2 parts, which is really the focus of our study. So the first is this direct effect of early life adversity on survival, independent of changing in social bond strength. And then the second is this mediation effect, which tells us how much of the effect of early life adversity on survival is due to this pathway through social bonds. So first, I'm going to tell you about how these relationships are changed when we consider female-female bond strength as the mediator. And so when female-female bonds are the mediator, we see a total effect of early life adversity of 1.5 years, which means that for every additional source of early life adversity a female experiences, her lifespan is decreased by 1.5 years. So a female that experienced 3 sources would have a lifespan reduced by 4.5 years. And much of this effect is explained by the direct effect of early life adversity in absence of the pathway through social bonds, which means that 90% of the effects of cumulative life adversity don't include a pathway that includes social bonds. We also see that cumulative adversity decreases the female's bond strength with other females, and independent of early adversity, if females have stronger bonds with females, they also tend to survive better. However, despite these effects of early adversity on female bonds and bonds on survival, we don't have a very strong mediation effect. In fact, mediation only explains about 1.6 months of the 1.5 years decrease in lifespan. So this means that only about 10% of the early life adversity, effective early life adversity on survival is explained by changing bonds with other females. When we look at social bonds with males, we see a similar story in terms of how much cumulative adversity affects survival, and that most of this effect is determined by a direct effect of early life adversity on survival in the absence of bonds. Interestingly, we don't see a significant effect of adversity on female-male bond strength, and this is likely because the variation is quite high. So some females tend to have pretty strong bonds when they experience more adversity, and some tend to have pretty weak bonds with males when they experience more adversity. We also see that in independent of early adversity, we have a strong effect of increasing bond strengths on survival. And although the mediation effect is of similar magnitude to models where female-female bonds were considered, there's no significant mediation effect when we think about a pathway that includes social bonds with males. So for both types of social bonds, we see weak evidence that they are actually mediating the relationship between between early adversity and survival. And so that really begs the question of, are social bonds important in adulthood? So we know that social bonds are important in fitness. Can they help mitigate the effects of cumulative adversity on survival? And so to answer this question, if they can reduce the negative effects of early life adversity on survival, we use the moderation analysis, which is analysis that tests for an interaction between cumulative adversity and social bonds in determining lifespan. And specifically, we predict that social bonds will have a larger effect on survival when females experience early adversity. And based on some literature in humans, we predict that adult females who experience early life adversity but are able to maintain stronger bonds will survive better than adults who experience early adversity but have weak bonds. So that these social bonds are important in overcoming early life adversity. So first, I'm going to show you a summary figure where I have social bonds with females on the left and social bonds with males on the right. I'm plotting each individual source of early adversity separately this time, as well as the cumulative adversity that we looked at and what I've described previously. And I'm showing you a hazard ratio interaction effect on the Y, which means that if there's positive values, early life social bonds with females amplify the negative effects of early adversity on survival. And if there's negative values, stronger social bonds reduce the negative effects of early life adversity on survival. And what we see for social bonds with females is there's no no interaction between the cumulative life adversity and social bonds on survival, but there is a reduction in the negative effects of early life adversity for individuals who experience maternal loss if they have strong social bonds with other females, suggesting that social bonds are really important in determining the relationship between adversity and survival. And we see a similar story with males. Here, we don't have an interaction effect for cumulative adversity, but we do for maternal loss. And in addition, social bonds with males seem to reduce the negative effects of early life adversity, in the particularly when their mothers are isolated socially. So I want to zoom in on maternal loss. And I want to also say that it makes sense that maternal loss might be buffered by social bonds in adulthood, because much of what you're losing when you lose your mother are social connections. A mom tends to be a female's strongest social connection, as well as her maternal relatives. So for individuals living without a mother, it might be really important for her to form bonds with other individuals. And so here I'm going to show you a reaction norm type plot where I'm showing whether an individual experiences maternal loss or not on the x-axis, and the hazard ratio on the y, where higher values of the hazard ratio mean a female is more likely to experience mortality and therefore has lower survival, and lower values indicate higher survival. So for social bonds with females, individuals who don't experience maternal loss have pretty similar survival probabilities, whether they have strong or weak bonds. But we see this interaction effect, a difference in slopes, when we consider individuals who experience maternal loss. Those with weak bonds have much higher hazard ratios and therefore are predicted to survive less long than those who have strong bonds. And for males, we see a similar story, very little difference with no maternal loss. And then for individuals who experience maternal loss, strong bonds with males are very important and can decrease or reduce the negative effects of maternal loss on survival. Okay, so to conclude, I've shown you today that early adversity and adult social bonds both affect fitness. However, there's really little evidence for a strong mediation effect. Social bonds with females, we found to weakly mediate the link between early life adversity and reduced lifespan, whereas we saw independent effects of early adversity and social bonds with males on survival. However, social bonds do play an important role in determining lifespan for individuals who lose their mother. Individuals who lose their mother but are able to maintain strong social bonds with males or females tend to survive better than those who lost their mother but cannot maintain strong social bonds. And as our results suggest that by considering this whole pathway, we can gain insights that weren't obvious from focusing on parts of the pathway. So from these studies that considered just parts of this pathway, we might have thought that there would be a strong mediation effect, but that's not what we see here. And we hypothesized that this weak indirect effects might be related to the timing of the effects across the life course. I don't have time to talk about that today, but I'm happy to share more about this exciting analysis later. So with that, I'd like to thank everyone who have helped collect the data, the Alberts Lab, our funding sources, and you all for listening. I'm happy to take any questions.
That was great, Liz. Thanks so much. We have a few questions in the chat. Does anyone want to ask their questions? Do we have any raised hands?
I can ask my question out loud.
Go ahead, Nicole.
Thank you, Liz. That was a wonderful talk.
Thanks.
So I was just curious. So based on some other work that you guys have done, I was wondering if you thought that perhaps glucocorticoids or something having to do with them could be a stronger mediator than either female-female or female-male bonds.
That is a great question. I'm also examining glucocorticoids as a mediator, and we find that similar to social bonds with males, it doesn't really seem to mediate the relationship. Instead, we think, like I said, that it could be due to timing of the effects. So the effect of early adversity on glucocorticoid levels and social bonds seems to occur is strongest later in life, whereas survival tends to be affected by factors earlier in life. So this timing mismatch might explain why we're not seeing a mediation effect. Good question, thanks.
That's fascinating, thanks.
All right, there seems to be one more question in the chat from Dr. Bernie Crespi, where he says the adversity.
Well, I can ask it, Joe, if you want.
Go ahead.
I couldn't find the raise hand. Yeah, it seems as though there's very, very different causes of variation in the adversity index, and some of them are quite non-social, and some of them are very social, and some of them seem to affect mainly the brain, and some of them will be affecting the body. So you did some analyses that looked at this, but I wondered if you did analyses that focused on the social adversities as opposed to the non-social adversities, or looked at interactions between them perhaps, or how they were related to one another. In that context, it seemed also that some of that the adversities could actually be inherited, and that you ought to have data on that. So things like social rank, for example, should tend to carry across generations. And it would seem as though the heritability of those effects could be quite important and relevant to humans as well.
That is a great question, and I have lots to say on it, but I'm going to try to keep my answer short, and we can talk more later in the breakout room. We find that maternal loss and drought are the strongest individual sources of adversity, but the results are pretty similar to the cumulative adversity. In terms of interacting effects, another project I'm working on is looking at interactions between individual sources of adversity, and we're not really finding a strong role of interaction. So that suggests that this additive index does a good job of showing the effects. But yeah, I'm happy to talk about more individual sources and socio versus physical environment sources later. Thank you.
All right, thank you, Dr. Lang. Now we will move on to our final speaker, Dr. Federica Pinarini. Completed her PhD in Germany at the Max Planck Institute for Evolutionary Biology. Her work was in the evolutionary immunogenetic group of Dr. Tobias Lenz, working on the evolution and function of adaptive immunogenic diversity in modern and ancient humans. After she joined the population genetics group of Flora J in Paris, there she has been working jointly with Emilia Huerta Sanchez from Brown University in the USA and Maria Avila Arcos from UNAM in Mexico on exploring the HLA evolutionary trends in admixed American populations. She's now moved back to Germany and at Max Planck Institute for the Science of Human History, where she's continuing her research on immunogene evolution through time and space in human populations.
Okay, thanks a lot for the introduction. I hope you can see my screen. And I would like to thank the organizer of this event series for the opportunity to present our work today. I'll be talking about our attempt to study a set of adaptive immune genes, the HLA genes in historical human populations. Just to give a brief background about the topic for those that are not familiar with HLA, HLA stands for human leukocyte antigen and is a gene-dense region of the genome which is located on chromosome 6. As you can see, it contains many, many genes, and most of those genes are involved in our adaptive immunity. Here you can see in colors the classical HLA genes, which encode for cell surface proteins, which are those in red, presenting on cell surface small peptides. When they present non-self peptides, for instance, pathogens peptides during infection, those can be recognized by the cells of our immunity. And so the adaptive immune response can start. HLA genes are known to be extremely polymorphic. Here you see the large number of different alleles that we know at the moment for each locus, and each dot here represents up to 50 variants. With the term allele, I'm not referring to single polymorphism, but in the HLA world, we refer actually to intragenic haplotype at each locus, so set of linked SNPs. And coming back to the HLA function, each haplotype encodes for slightly different molecules having different binding properties towards pathogens peptides. So because of this important role in our adaptive immunity, the large diversity that we see at the HLA genes has been proposed to be the results of the selective pressure imposed by the diversity and coevolution of pathogens. And according to this notion, different mechanisms of pathogen-mediated selection have been proposed, mainly the heterozygote advantage, the negative frequency-dependent selection, and fluctuating selection. All these mechanisms work in parallel, but on different time scales. And basically, those mechanisms have been extensively studied in other species and also in humans, mainly focusing on the genetic diversity at HLA in modern humans population. But what remains still unexplored are those patterns in historical humans population, so basically ancient data set. So basically to explore ancient DNA data and explore genetic diversity at HLA might help us to identify allele frequent changes over time, and so to better explain the differential pattern of pathogen-mediated selection. However, to genotype HLA genes using ancient DNA is extremely challenging. First, because of the high density of SNPs that we found in the peptide binding domains, which are this region in red, which are the region we need for to genotype the HLA genes, and for the high level of paralogous sequence similarity along the full HLA genomic region, and also for several features of the ancient DNA samples, which are often highly degraded. There is high risk of contamination, and also the DNA is always sometimes present in small quantities. To overcome those problems here, we applied a specific HLA capture approach together with a pipeline that we call TARC pipeline. And this method was already applied to different data set of ancient samples. And I will talk today about two of these studies coming back from two different historical contexts. The first one goes back to the medieval time, and is a data set of medieval people who died from leprosy. So leprosy is a chronic infection disease, which is caused by the Mycobacterium leprae, and in its severe form can leave diagnostic markers on the skeletal remains, both in cranial and postcranial bones. Leprosy was endemic in Europe during the medieval time, but disappeared after the 16th century, and nowadays is absent in Europe, but still present in more than 100 countries. A strong association has been reported with the HLA region, especially with the DRB1 1501 allele, which is known to be a risk factor in different human populations, for instance, from Brazil, China, and India. So in this work, we applied our HLA capture approach to 69 samples. The DNA was extracted from skeletal remains that show the typical leprosy lesion, and when collected from a leprosarium in Denmark, here the red dot on the map, and we were able to reconstruct the HLA profile. So what you see here is the percentage of allele codes, the different classical HLA loci, which is quite variable. And this was basically depending on the quality of each samples and the coverage we could reach for the HLA region. The HLA allele code were possible at two different levels of resolution. The first field level, which help identifying the general serological type, and the second field level that can distinguish alleles sharing similar functional properties, but still differing in their protein sequences. We next compare the cases with more than hundreds control. which were basically samples collected from ordinary symmetries in the same geographical region and from the same period, which are the black dot instead. The controls were screened for the presence of the allele DRB1 1501 using a tag SNP, which is a SNP known to be in linkage with the variant we are interested in. And so we can see here a significant association between the cases, the ancient cases, and the variant, which suggests that the DRB1 1501 was also predisposing medieval European to leprosy. We next explore the binding properties of the different DRB1 molecules using peptide binding prediction softwares. And what you see here are the number of Mycobacterium leprae, which are predicted to be bound through the software by the different molecules. With our variant here, the 1501 in red, showing a limited presentation of Mycobacterium leprae antigens, which can give us an interpretation of why this variant is indeed a risk variant. We now go further back in time, and we focus here on the Waterberg culture, which dates back to the late Neolithic period. The archaeological remains from which the DNA was extracted here come from a collective grave located in central Germany in the state of Hesse. And this collective grave was likely used by the same community over hundreds of years. And what we obtained first are genome-wide data from 42 individuals. And you can see here a PCA plot where we compare the variability of our late Neolithic population in red with ancient samples coming from more than 100 different populations, basically. And the cluster can be mainly explained, we see, from by the genetic diversity of early hunter-gatherers and early farmers. What was surprising about this late Neolithic population in red is also the high amount of hunter-gatherers ancestry, which would be better seen from the admixture plot. Our population is the one in the red rectangle, and we see there are two main ancestry components. The blue one, which are the Neolithic Anatolian farmers, and these account for more or less 60% on average, while the hunter-gatherers component account on average for 40%, so quite high. We next applied our HLA capture approach to 23 unrelated individuals, and we described the HLA repertoire in the ancient dataset. Here we see the allele frequency in the ancient dataset, which were then compared with the allele frequency in modern German individuals. And for the two most common allele in the ancient dataset, we can see a substantial change in the allele frequencies when compared to modern time, with 9% of the diversity, at least, differences. So for 7 of these most common alleles, we were also able to use, again, tag SNPs to trace allele frequency changes over time, basically looking at this variant in a large data set of European ancient samples that were basically taken from previously published work. And we can see that 5 of these 7 variants have a high frequency in the hunter-gatherers population and a reduction over time until modern day. But the frequency is still quite high in our late Neolithic population, which indeed reflect the large amount of hunter-gatherers ancestry component that we saw previously. So some of these allele frequency differences between epochs were significant. And I will now focus on one of these trajectory, which is the one from the allele 0102. Again, here we see a comparison in the allele frequencies between ancient and modern individuals, with the 0102 allele here being one of the most frequent in the ancient dataset and showing a significant reduction with modern day data. And to further explore this pattern, we here have a phylogenetic relationship between 11 ancient HLA-C alleles with the 0102 showing a higher sequence divergence to the others. But why having such a large sequence divergence might be advantageous in functional terms, I will try to briefly to explain. So basically, if we think at the genotype level, individual having being homozygote, they have same allele at a given locus, and in functional term, the molecules will bind the same range of antigens. Heterozygote individuals with similar alleles will be able to bind a larger range of antigens than homozygote, while heterozygote individuals having more divergent alleles at the genotype, so larger number of differences along the sequences, will be able to bind an even larger range of antigens. And so this large sequence divergence increase the number of bound antigens, and this to translate into higher immunocompetence. Keeping in mind this assumption, we next compare basically genotypes in the ancient data set carrying and not carrying our allele, the 0, 1, 0, 2. And we saw that indeed the carriers show higher sequence divergence in this case. So this can give also an explanation why this variant could be advantageous in ancient time, and so why the frequency was higher. To sum up, we saw the potential of exploring HLA allelic diversity in ancient data sets, so basically to understand better the differential mechanism of pathogen-mediated selection through time. We saw an association between leprosy and HLA variability in ancient medieval samples, and we saw also a drastic change in the HLA profile over time in Europe from the hunter-gatherers population until modern time, which suggested that the HLA repertoire of modern Europeans was established quite recently, likely during the last 5,000 years, and was likely affected by the admixture process. Let me just acknowledge all the collaborators that were involved in this project, and also who lead the project. So my PhD advisor, Tobias Lenz, and a special thanks to Almut and Ben, and also to Alexander and Marcel, and to all of you for your attention.
Thank you, Federica. I'm looking in the chat to see if I can see any questions. We've got about 2 minutes. I'm going to ask one quickly because I don't see any. So what jumps out at me in terms of the difference in HLA diversity between the hunter-gatherers and the farmers is that the farmers were probably living in higher density populations and therefore more exposed to transmission of pathogens. So do you think that played a role in the change that you saw?
Yeah, of course. One of the hypotheses is indeed that the pathogen landscape had a drastic change during this transition, and it might be that there was really a replacement of pathogens, likely. So one of the hypotheses is that viral pathogens were more frequent during the hunter-gatherers period, while probably bacteria were more prevalent in the later time. But of course, we need to explore this further. Maybe also ancient pathogen sequence will help to understand this better. Yeah.
If I could just ask, what was the hunter-gatherer population and where were they located? Sorry if I missed that.
And they do, basically we compare the previously published work. Now I cannot tell you specifically the name of the population, but we used all the published hunter-gatherers from Europe, basically.
Okay, from Europe.
Yeah.
Okay, thanks.
All right, Dr. Bernie.
Probably the biggest pathogen-related selective event in human history that we know of is, of course, the Black Death. And we have all these wonderful, convenient graveyards full of the Black Death victims. I mean, I don't mean to suggest, that you should go in there and dig them up yourself, but it seems like a wonderful opportunity that caused major changes in the genetic structure of Europeans across the board. Do you think that that's a feasible project, or you thought about that?
Yeah, we are thinking to explore this, and actually, yeah, so far I think other people tried, and we still didn't observe any drastic change in the HLA. But I think yes, we want to explore this further, and yes. Yeah, because also the mortality rate was very high. And so basically the signal there might be stronger, yes.
We have time for one quick question. Nicole, would you like to voice your question? Because I want to hear an answer to this one too.
Yeah, sure. So thank you, Federica. That was a wonderful talk and analysis you've done. I was wondering if you could venture to comment about European HLA diversity, either being influenced by colonialism or somehow influencing colonialism.
You mean American colonialism or in general?
Yeah, they got around. So
okay.
Europeans everywhere.
Okay, yes, of course, that will be great. And I think for this, I would love to basically to continue exploring ancestry patterns together with the change of HLA frequencies, right? So in this, I think there's really need to combine the two fields, so to see how the patterns of admixture and ancestry have been changed along history together with the changes in the HLA region. And I think maybe, I don't know, one thing that I would love to do is to define local ancestry patterns in the HLA to check how indeed the admixture migration from European into the European population may have affected HLA, yes.
I mean, one thing that you might want to look at, because we have excellent historical narratives of epidemic diseases in European colonists in Africa, and a lot of those colonists stayed there. And so their descendants still live in Africa, and they are, of course, descended from people in the Netherlands and Portugal and France. So that would be a really interesting study to see whether there was selection on those European HLAs who were able to stay in Africa after the colonization events compared to the host countries they came from.
Yeah, one thing that we were exploring also with the other supervisor, Flora J. and Emilia and Maria, it's like the increase of African ancestry in the admixed American population, which is kind of similar story, but in the other continent. So basically, it seems that there is an increase of African ancestry in the HLA, in the admixed American population. So yeah, maybe the other way in the African would be really interesting, yes.